Hello everyone, welcome back to my channel. Thank you for tuning in for another exciting video today. So in this video, we are going to continue the discussion from the previous video, where in the previous video we learned about what is single cell ATAC-Seq data, how do we process it, how do we interpret it. So today I'm going to continue with the same data, and today we are going to learn how to perform some downstream analysis on the pre-processed single cell ATAC-Seq data. So if you haven't watched the previous video, please make sure you check it out. I will add the link to it in the description section below. If you have watched the previous video, we have previously discussed about the single cell ATAC-Seq analysis workflow. Um, Quickly giving an overview, this workflow is broadly divided into pre-processing steps and downstream analysis. And in the last video, we focused on the pre-processing steps. We also learned how to pre-process single cell ATAC-Seq data. But today we are going to be focusing on the second half of this workflow on the downstream analysis, where we will be taking the pre-processed data, and the first step that we will be performing is the cell identity annotation. We will be identifying the clusters, and there are various strategies to perform cell annotation in single cell ATAC data, which I'll be talking about in a little more detail in the next slide. And followed by that, we will be performing differential accessibility analysis with our identified clusters to identify which are the regions that are differentially accessible between certain cell types. Followed by that, we will also be looking at how to visualize the genomic regions of our interest with our processed and labeled single cell ATAC-Seq data. There is a really informative article by 10x Genomics that talks about various strategies of cell annotation, which is what I referred to when I was putting together this slide, and I will be linking that article in the description section below. So they talk about the three main strategies to annotate single cell ATAC-Seq data. So the first one is annotating using regulatory elements. The idea here is that cells are labeled by visualizing the promoter accessibility patterns of cell type marker genes. So the idea here is there is an assumption that there is a general correspondence between gene body or promoter accessibility and gene expression, which may not always be the case, but we assume that if there is accessibility in the gene body or promoter region of a gene, then that gene is expressed in that cell. So if there is a high expression of a marker gene in a cell, then there are higher chances of the cell being of that particular cell type. And this will be demonstrated when I walk you through how we go ahead with the cell annotation. So this will be clear, but we will not be utilizing this strategy to annotate our single cell ATAC-Seq data. So the next strategy is annotation using cell type specific feature set. So the idea here is to compare the enrichment of certain features. These could be DNA peaks, gene activation scores, or motif accessibility, comparing it against the background levels and finding out which of the cell types has highest enrichment for that particular feature set and labeling that cell type. So to give you further idea about this strategy, let's say we identify top 200 peaks that were most enriched in each cell type, and we compare this enrichment with the background to make sure that these identified top 200 peaks are truly specific for each cell type. And then we look for the enrichment of this peak in all the peaks set in all the cells and identifying the cells with highest enrichment. The cells with the highest enrichment is of that cell type. And the third strategy is annotation using RNA sequencing data as reference. So basically performing cell type predictions based on distances to the cells that are pre-annotated by RNA sequencing data. And this is the strategy that we will be using in our demonstration today to annotate our cell types in our single cell ATAC-Seq data. So the data for today's demonstration is the same data set that I used in my previous video, which is a single cell ATAC sequencing data set of human peripheral blood mononuclear cells by 10x Genomics. And the goals for today's demonstration is to annotate cell types by integrating with a pre-annotated single cell RNA sequencing data, finding differential accessible regions between cell types, and visualizing genomic regions. So these are the requirements for today's demonstration. We will be utilizing scATAC to process single cell ATAC-Seq data, sRAT for integrating with single cell RNA sequencing data, and tidyverse to perform data manipulations and wrangling. So for this demonstration, we will start with pre-processed single-cell ATAC-Seq data that we pre-processed in the previous video. We'll be using the same data. So if you haven't watched the previous video, I will be adding the link to that in the description section below. So for the previous video as well as for this video, we will be following their official vignette. So again, I will be adding the link to this in the description section as well. So in the previous video, we had performed until nonlinear dimensionality reduction and we had performed clustering. So what we have right now is pre-processed single-cell ATAC data with these clusters which are not identified yet. So in this video, we'll continue from this point onwards with identifying clusters and performing differential accessible peaks between different cell types and also visualizing the genomic regions. So let us first start by loading our libraries first. And this is the pre-processed single-cell ATAC data that we pre-processed in our previous video. So continuing with the same object and just quickly plotting a UMAP with clusters. So basically to show you the clusters that are identified that are not yet labeled. So we have identified certain number of clusters, but we haven't identified what these clusters are. So let us first start by creating something called as a gene activity matrix in order to integrate with single cell RNA-Seq data so that we will be able to integrate them and identify what these clusters are or what these cell types are. So the direct comparison of chromatin accessibility data with gene expression is challenging due to their different natures. As single-cell RNA-seq, we have seen the feature barcode matrix where features are the genes and we have the counts which gives us the quantification on the expression of these genes in different cells. Whereas in single-cell ATAC data, what we have is regions. We have the regions where we find the chromatin to be open in different cells. So in order to make direct comparisons and to integrate these different modalities together, what we need is to get the single cell ATAC data into a format that is more comparable to single cell RNA sequencing. So the central idea here is to identify shared correlation patterns in gene activity matrix and single cell RNA sequencing dataset to identify matched biological states across different modalities. So this matrix provides a way to approximate which genes are being actively transcribed based on the accessibility of their regulatory regions. So the general assumption here is the open chromatin means that the genes are being actively transcribed, which means that the genes are being expressed. So to give you an example, if a particular cell has open chromatin region in the promoter and enhancer region of the gene MS4A1, then the gene activity would reflect high activity for MS4A1 in that cell, suggesting that MS4A1 is likely being actively transcribed. So gene activity matrix is created by intersecting the fragments with the gene body and the promoter regions for each gene and then summing those fragments for each gene. So there is a function that will calculate the gene activities for us and we can store this in another object. And I can quickly show you what that would look like. So now this is finished running. Let me quickly show you what this looks like. So this is going to be a sparse matrix, just like how we had a feature barcode matrix in our single cell RNA sequencing data. So here we have the open fragments that are overlapping, the sum of the open fragments that are overlapping the the open promoter and the enhancer regions. So now let us store this gene activity matrix into another assay in our object, because this would be directly compared to the single cell RNA sequencing data that we are integrating. So before we do that, we would have to normalize and scale this data in order to compare and integrate them. So we are going to store it in another assay called RNA. So we are going to create assay object and the counts are going to be the gene activity matrix. So now in addition to the single cell ATAC-Seq, we now have another assay called RNA where we have stored our gene activity matrix. So let us now normalize and scale this data. So we are going to use normalize data function. The object is going to be PBMC. The assay is going to be RNA. The normalization method is going to be log normalize. And for scale factor, we are going to be using the median of the counts. The scale factor represents a global scaling factor. They adjust the raw counts prior to the log transformation, and here choosing the median value as it's more robust to outliers and is a more representative central value for scaling the counts across the cells. So let us save it back to the object. So now that we have our gene activities normalized, let us try to interpret some of the single cell ATAC-Seq clusters by visualizing the activity of some of the canonical markers. Since we know that these are peripheral blood mononuclear cells, we have a basic or a fair idea of what types of cells we would expect to find in our data. So we could visualize the activity of these canonical markers, which would be a way of interpreting it as the expression, because since these are open fragments which are overlapping with the open promoter and the enhancer region, the regulatory region, it can be assumed that these are being expressed since these are more accessible for transcription. So let us first set the default assay of the object as RNA, since this assay holds our gene activity matrix. And let us create a feature plot. And in this features, we add some of the canonical markers that we would want to visualize. So I'm just going to type up the name of some of the canonical markers. And again, I'm following the one that they have mentioned in their vignette. But if you're trying to do this with your own data, this is one of the strategies to annotate your cell types if you're well aware of the cell types that are present in your data. So this feature plus shows different clusters showing higher gene activities for some of these markers, which indicates that these are the markers that have high expression in these cell types or these clusters, and that could help us identifying which cell types these could be, since these are all canonical markers. So as I said, this could be one way of annotating the cell types if you have a good knowledge about the cell types that you expect to be present in your data set. However, we will proceed with integrating with the single cell RNA sequencing data, pre-annotated and pre-processed single cell RNA-seq data, and we would be using that to perform label transfer and identify the cell types in our single cell ATAC data. So in order to integrate with single cell ATAC data and to perform label transfer, we are using single cell RNA sequencing data from the same system, that is human PBMCs. So in the vignette, they have provided a link to pre-processed single cell RNA sequencing data. So I'm just going to read that data in. I've already downloaded that data. This is the link to that data. Again, I will be adding this link in the description section as well. But you could download this data. This is pre-processed, so you don't have to perform any pre-processing on single-cell RNA sequencing data. If you are working with your own data set, then make sure you pre-process your single-cell RNA sequencing data first before you integrate. So I will read that data in here. So taking a look at this data, it says that we would have to run update Seurat object on this object. So let us do that. Save it back to. So here is our single cell ATAC-Seq data with so many features and so many cells. So let us take a look at the metadata. As this is supposed to be already annotated, Just want to make sure that we have cell annotations present here for each cell type. So let us plot these annotations, just visualize these clusters before we integrate. Let us visualize our ATAC clusters and our single cell RNA-Seq clusters side by side. And saving that to a variable so we can visualize them side by side. So doing the same for the RNA-Seq as well. So our annotations are present in the column cell type. So we want to group it by cell type. We want to show the labels as well. Let us save this into another variable. So visualizing both the datasets side by side, we have been provided with a pre-processed and annotated single-cell RNA sequencing data from the same system, and we can see here that the clusters have been identified, the cells have been labeled. So this can be used as a reference data to transfer the cells into our query data, that is the single cell ATAC-Seq data. And we know that these share the same cell types because they are from the same human system. They're both from the human PBMCs. So we know that we can use this data set to perform label transfer and identify clusters in our query data set. So we will be using Sirath's methods to identify anchors and perform label transfer. So the reference here is PBMC RNA. The query is going to be our PBMC, which is our ATAC data. And the reduction that I'm going to use is PCA project. I'm not using CCA because CCA is very slow for me. So I'm switching to other methods. And here I'm going to save it back into an object called transfer anchors. The transfer anchors contains the pairs of cells that are very similar between the two data sets, and that will be used to transfer the labels from the reference to the query. So now that we've identified the labels, or rather the anchors, let us use these anchors to perform the label transfer. Let us provide the anchor set that we identified. This is the labels that have to be transferred. So the labels are stored in the PBMC RNA in the cell type column in the metadata. The weight reduction for the query will be LSI. This is the weight reduction for the single cell ATAC data. And we use this method to perform dimensionality reduction in our pre-processing. So if you haven't watched the previous video, we have spoken about this in the previous video. So make sure you check that out. And we will be considering these dimensions. Let us store this in a variable called predicted labels. So let us take a look at the predicted labels. So the predicted labels provide for each of the cells, it provides a score for each label that is present in the reference data. And the score is an indication of how confidently the cell is assigned to a particular cell type in the query data set based on the reference data. So higher the score, higher is the confidence of that cell belonging to that particular cell type. So let us add this information into our ATAC data. And let us take a look at the metadata of the ATAC-Seq data now. So here we can see that in addition to the metadata columns that were present, we have the predicted IDs and we have the scores for each of these labels for each cell type. Now that we have performed label transfer and we have predicted cell types for our single cell ATAC-Seq data, let us visualize both the data sets together again. So let us create dim plots. Let us store this in a variable called plot1. I'm going to copy the same and change certain things for RNA-Seq. So let us run both of them now and plot them side by side. So now visualizing both the data side by side and having labels on the single cell ATAC data, I can see that it has done a fair job in identifying cell types in the clusters. However, there are certain inconsistencies that I see that I don't expect CD4 to cluster with CD14 and 16 monocytes. I expect to see the cells, CD4 naive cells to be somewhere closer to CD4 memory cells. And the only thing I think I have done differently from the vignette is the reduction method while identifying anchors. That is, I have used PCA project instead of the CCA, which has been used in the vignette. That could be one of the reason. But if I were to repeat this or do this on for my experiment, I would perform multiple. I would use multiple methods to perform label transfer, and at the end use some sort of consensus to amongst the labels that are identified from the different methods to identify the clusters. But for now, I think we are going to run with this and we are going to move ahead with performing differential accessibility analysis between different or certain cell types. So in order to perform the differential accessibility analysis, we need to set the identities of the cell types in our ATAC data to be the predicted cell types. So right now the identities are the clusters. So each cell belonging to a certain cluster. So we need to change this to the cell types that are predicted. So we set this to the predicted cell type. So running the identities again, we see that now instead of the cluster number, we get to see what cell type does that particular cell barcode belong to, or what that particular cell belong to. Now, in order to identify the differential accessibility between peaks, we need to change the assay from RNA to ATAC, because remember our active assay is set to RNA. So we need to change this to the ATAC because we need to switch to peaks from the gene activities. So let us set the default assay to ATAC. And now when we check the default assay, the default assay is ATAC. So now let us find differential accessibility between certain cell types. So for that, we use the function find markers. We provide the object. We provide which cell type, which two cell types that we want to compare. So you want to compare CD4 naive and CD14 monocytes. We are using the number of open fragments, the counts of that for ATAC, and let us store this in another variable called DA peaks, that is differentially accessible peaks. This is Siraj's find markers function, and previously I have created a video on find markers function from Siraj in addition to other functions like find all markers and find conserved markers, and make comparisons between these functions, explaining how these functions work and which function to use when. So I will be adding the link to that video as well in the description section below. So now this has finished running. Let us take a look at the differential accessible peaks. So essentially what we get is these differentially accessible regions between the two groups of cells that we are comparing. So we have these regions, the p-values, the fold changes, the percentage of accessibility in these groups, and the p-adjusted values. So the way that we interpret these regions as, for example, this region has differential accessibility, and this is the fold change. It is these many times more accessible in one group compared to the others. So now let us visualize the first region, that is the most differentially accessible region. So for that, let's create a violin plot to compare the accessibility between the two groups of cells. So the object here is going to be VBMC. The features is going to be first region, setting some aesthetics. And let's store this in a variable called daplot1. Now let us create a feature plot. And let's store this in another variable called daplot2. And now let us visualize them side by side. So here we can visualize it two ways, as Violin plots and as feature plots, to see the regions that are differentially accessible between the two groups of cells that we are comparing. Now, if we want to identify the peaks or the regions that have a very high fold change of accessibility between the two groups of cells, then there is a function that we can use called the fold change, which will allow you to get the fold changes for the regions that you're comparing between the two groups of cells. So here, let's say we are comparing CD4 naive and CD14 monocytes. So this essentially extracts the fold change for the regions that have differential accessibility between these groups. And now you can also order the fold change by the, you can also order these fold changes by the highest fold changes by increasing or decreasing value. So we want the highest fold changes at the top. So this gives you, let's just do head. So this gives you the regions with the highest fold changes between the two groups of cells. This helps you to narrow down certain regions that you may want to inspect further or look into further. So this is one way to extract these regions with the highest fold changes. So now coming to the last aspect of the demonstration, that is to visualize the genomic regions of your interest. So let's say if you want to visualize the differentially accessible regions across all your cells or clusters, then the first thing we need to do is to set the levels for plotting. And this should be the predicted IDs. Do the unique to that. So essentially your levels are basically setting these levels for the plotting. And followed by that, we have a function called coverage plot, which allows us to plot the region of our interest. So the region here, I'm going to choose the first region that was identified as differentially accessible in the differentially accessible peak object. Then you can extend the regions upstream or downstream, because a lot of times it is helpful to visualize certain, let's say if you're trying to visualize a region, a gene region, then it might be of interest to visualize certain bases upstream to get to know the accessibility in the promoter or the enhancer region, because those would give you an idea whether the gene is being transcribed in one cell or one group of cell versus the other. So it allows you to extend upstream. Let's say we extend it by 40,000 bases and extend downstream by 20,000 base pairs. And now let's create or visualize this plot. So here in this plot, you can see that the first region that was identified as differentially accessible is more accessible in certain cells compared to the others, is more open in or more accessible in CD4 memory, NK cells, CD4 naive cells, double negative T cells, but not so much in B cell progenitors, CD16 monocytes, dendritic cells, pre-B cells. So this is a useful way of visualizing your regions of interest to see which of the cells have more open chromatin, and which means that which of the cells, if that region corresponds to a gene region, it could also give you an indication of which of the genes are being more open and expressed in one type of cell versus the other. At the bottom, you'll also get to see the gene and the peaks. Notice the gene is on the negative strand as the arrows are pointing to the left. So that would make this region to be the promoter and the regulatory or the enhancer region. So you would essentially be interested in looking at which of the regions are more accessible in terms of like promoters and enhancers for this particular gene if this is your gene of interest. Lastly, you can also create an interactive browser to visualize your regions. So to do that, there is a function called coverage browser where you provide your object name and you provide the region. So let's say we provide a gene region CD8A and run this. So here it creates a small shiny application which can be opened using this small icon that is shown in a new window. So here you can explore different regions by using these arrows going left or right. You can also change or change the chromosome, the regions here. You can also change the aesthetics in terms of what you want to display in this plot. You can also plot gene expression side by side. So let us change the gene to MS4A1. And let us also visualize the expression for the same gene. So you can visualize the accessibility and gene expression side by side, and you can click on update plot. It takes a couple of seconds for the plot to be updated. So when you go to the view tab here, you will be able to see the accessibility for this gene in all your cells corresponding with the expression. So here you can see that this region has more open chromatin or accessibility in B cell progenitor and pre-B cells. Makes sense because MS4A1 is a canonical marker for B cells, and you can also see the expression for these for this marker in different types of cells. You can also go left and right, upstream or downstream in this plot using these arrows. Whenever you click these plots, it takes a couple of seconds for the plot to update. But again, this can be a very useful tool if you want to explore different regions in your data and you don't want to go on recreating different plots. This could be a very handy way of exploring different regions and assessing the differential accessibility of these regions in your cells. So that brings me to the end of this video. I hope you found this video useful and informative. You will find the links to all the resources and all the videos, references that I made in the description section. You will also find the link to my GitHub repository where I host all my code. So you will be able to recreate the same analysis. So if you enjoyed this video, please consider giving it a thumbs up. Don't forget to subscribe to my channel for more bioinformatics related content. If you have a question or a suggestion for a topic, please leave it in the comment section below. Share this video with your friends and co-workers. Please stay tuned for the next video where we will be exploring more of different bioinformatics tools and pipelines. I really appreciate your support and I'll see you in the next video.